You just either saw the transparency. Anyway, it's my pleasure to introduce Natalie DelRocco, joining us from the University of Florida in Gainesville. She's a graduate student there and is here interviewing for a faculty position here at SC and working with the Children's Oncology Group. And her talk is on 2 contributions to statistical improvement of clinical research. All yours.
Thank you, Todd, for the introduction. Hi, everyone. My name is Natalie. It's great to see you all here. Thank you for attending. I am so excited to share 2 of my research projects that have been contributing to statistical improvement of clinical research. There we go. So I'm going to start out by discussing a newly proposed method for extreme outcome-dependent sampling. Louder?
If you move the microphone, he's saying if you move the microphone, then see if
This one? This microphone? And yeah, I can talk louder.
Try that.
Did that work? It's better? Okay. All right. Just do this. Okay. So yes, extreme outcome dependent sampling estimator. And then I'll transition into a prediction modeling project that is aimed at enhancing risk stratification for pediatric acute lymphoblastic leukemia. I'll briefly touch on my future research plans, and please do interrupt me if you have any questions during this talk. I was thinking I would save like 15 minutes at the end for questions, but I think I still have to wrap up by 1 and it's 1:15. So we'll see how that turns out. So the first project was motivated by the BISP2 clinical trial. It was a phase 2 clinical trial looking at the effect of pharmaceutical and educational interventions on induced shoulder pain. So importantly, the primary analysis found no significant difference between the treatment and the placebo arms. So what my project was focused on was the exploratory biomarker analysis following the primary analysis. So they were interested in knowing the associations between biomarkers of interest and continuous worst shoulder pain, which was measured using something called the Brief Pain Index. So there's no universally accepted definition of biomarker, but for the rest of this presentation, I'm using biomarker to refer to a continuous concentration of a substance that's measured as a part of a serum sample. So something in your blood. The most familiar one to you might be cortisol. So why is this challenging? It looks pretty standard. Well, the pandemic hit. It really threw us a wrench. We had a huge financial constraint. No one was enrolling in the primary trial. And so the primary trial got dragged out and dragged out. There were serious concerns that we weren't going to have any funding for the biomarker analysis. And actually, bigger than the financial constraint ended up being the time constraint. So where previously it might take you maybe a month to get your biomarker assays from the lab, there was 1 scientist working at a lab that was meant for 5 scientists on a rotating schedule. Everyone was getting sick. It took us 11 months to get a preliminary biomarker assays done. So what that ended up meaning is that we didn't have the time or the money to biomarker type our full sample plan. So a potential solution might be to be intentional about who you're selecting for biomarker typing. So this might sound kind of familiar to you guys. The inspiration comes from optimal design. So you can think of a de-optimal design. The solution for linear regression maximizes the information in the Fisher information matrix, and that would equate to the top and bottom most extreme individuals that you can get in your sample. That's what would give you the most information for that optimal design. The idea of selecting extreme individuals also shows up in a topic called selective genotyping. So in selective genotyping, we typically have a continuous phenotype and a binary genotype. And what geneticists will do is they'll specify a most often they'll specify a full or a conditional likelihood function that is highly specialized to the genetic application that they're working on. So it might incorporate something like genetic backcross, and it ends up not being applicable in general settings. By the way, there are so many more details in this current literature slide. I'm happy to powwow after this if you have questions. Selective genotyping generally falls under the outcome-dependent sampling paradigm. So outcome-dependent sampling, though, traditionally, most often does not have an extreme sample from the top and bottom tails, but they'll have a simple random sample from the full support of your response variable, and then additional simple random samples in areas of the support that are most interesting. So like the upper and the lower tail, similar to an extreme sampling scheme. So just to visualize the challenge, we have a continuous response variable. So that would be Y here. And then I have a chat. Oh, no.
Sorry. We're having more technical difficulties,
no.
everybody. Sorry. I'm going to stop share real quick.
Oh, no. That has been me before. Okay,
Let me just know. Okay, I
good.
think we're good now.
Okay. So we have a continuous response Y and a continuous expensive covariate X. So if we're pretending that I am able to observe all of the Ys and all of the X's, here's what I would get if I was looking at an association between X and Y. So if I fit a simple linear regression to the full population, I get the solid black line. So the challenge that we were trying to address is, well, now if we select the top and bottom most extreme individuals on their Y value, how can we analyze that without bias? And as you guys might expect, too far. As you might expect, just fitting an OLS model to the extreme sampled points does not work. It severely biases your estimation. You are not recovering your regression line that you would have gotten if you were able to biomarker type all of the individuals in your sample. And so the reason that this occurs is the conditional distribution of Y given a particular value of X that the simple linear regression is depending on no longer follows that normal distribution that it needs, but it is now a truncated normal distribution. So that biases the line. What we need is a method that recovers closely the black line while only relying on the red points and using the full information from Y, because remember, we have all of the Ys, we just can't afford to collect all of the X's. So the intuition behind our proposed solution is, what if instead of looking at the traditional Y regressed on X regression, we looked at X regressed on Y? So the distributions, the conditional distributions that are important in that case are the conditional distributions of X given a particular value of Y, which is preserved if we conduct the selection on Y. So they're untouched and truncated. So I'm briefly walking through the estimation. I'm hand waving over a lot of algebra details, and I have a preprint of our paper, which is currently under review, that I'm happy to share if you're interested in the algebra. But here we're making a big parametric assumption. So of course, this is the exception, not the rule, and you have to be very careful with it. We're assuming that X and Y jointly follow a bivariate normal distribution. So as we discussed, the traditional linear regression equation is no longer valid, but we're still interested in recovering this beta Y from the regression. So the reverse regression, X given Y, is valid, and we can use it to solve for that beta Y of interest. We're assuming the standard normal with mean zero and constant variance errors in this regression equation here. So using this familiar formulation of the bivariate normal distribution, and if you don't recognize it, because it's probably been a long, long time, don't worry, you just need to know that this is where the bivariate normal assumption comes in. So we are, without loss of generality, plugging in standardized X and standardized Y into this theorem, and then we can solve, and this is the algebra hand waving part, we can just solve for the parameters that we're interested in. And again, asking you to trust me here, but here's what it works out to. So beta Y can be solved for in terms of beta X, the error variance of the reverse regression equation, and the full variance of Y. So beta X and the error variance of X are standard outputs from your statistical software that you would use to fit your reverse regression equation. And the variance of Y, of course, you can use the sample variance of Y because we have all of the Y observations. So plugging in estimates for beta X for the error variance and for the variance of Y, which we're denoting sigma tilde Y, just as a reminder that that's unique. We're using the full information from the Ys, whereas for the other ones, we're only using the extreme sample points. We're calling it beta hat ODEB, outcome dependent extreme biomarker typing, just to kind of get rid of the confusion with the X's and Y's. So this is our proposed estimator. So we can also derive an expression for the standard error of beta hat using the delta method. These are all the quantities that we've already discussed in a standard that come from fitting the reverse regression model. And S here is the number of individuals that you actually sample, whereas NF is the number available to sample. And the corresponding 1 minus alpha % confidence interval can take the usual form. So we looked at a whole bunch of simulation settings. I'm giving you the highlights here, just the most important piece. We considered a single continuous biomarker X. And as I mentioned, we're assuming that the errors are normally distributed with mean 0 and constant variance. We considered a few different sample sizes. Sizes 100 to 400 are what we would typically see in that type of clinical trial that we were working on, with 800 just showing what the characteristics of the estimator were under a large sample size. We constructed a continuous model for pain, and we tested a range of betas that were appropriate under the biomarker situation, according to our clinical collaborators. So for each iteration, we conducted outcome-dependent sampling and random sampling, and we looked at various proportions sampled. So 10% from the total sample, which would be quite a small proportion, 20%, and then 40%, which would be a large proportion. For each iteration, then we calculated the two different estimators and compared the performance. So as you can see, the blue indicates OLS estimation. We already discussed that we're expecting that to perform poorly, while red indicates the new proposed estimator. The dashed line is random sampling, and the solid line is extreme sampling. Hopefully the colors are okay to see. But what we can see is that the blue solid line, which indicates OLS estimation under extreme sampling, has severely inflated bias, while all of the other situations concentrate the bias around 0. So the new estimator keeps the bias low. Similarly, for the RMSE, under extreme sampling for OLS estimation, we have very high RMSE, followed by the new estimator and OLS estimation under random sampling. And then our new estimator under extreme sampling actually provides you a lower RMSE because you're using all of those Y values, whereas these OLS regressions are only using the extreme sample points. So you're buying a little bit of estimation accuracy there. For the power, under any circumstance, the type 1 error rate is controlled, but I think what's really interesting is the relationship between the proportion that you've sampled and the sample size. So the black stars in the top right panel denote 80 participants sampled. Each black star is 80 participants sampled. The red line is 10% sampled. The green line is 20% sampled, and the blue line is 40% sampled. And so what this is showing us is that a sample of 10% from a full population, a trial population of 800, has more power than a sample of 20% from a size of 400, which buys you a little more power than a sample of 40% from a size of 200. So the more extreme your sample, the more power that you're gaining. And the confidence interval coverage, as we might expect, deteriorates very quickly for OLS estimation, while the ODEB estimation maintains nominal coverage around 95%. So in summary, when assumptions are met, you can have increased power, particularly for a small sampling proportion from a large available population, while preserving the type 1 error rate. And you buy yourself a little bit of estimation accuracy, it lowers the MSE. The limitations are the strong normality assumption. So this is a strong parametric model. We do have simulations showing that it is quite susceptible to heavy-tailed distributions. The outliers that are popping up are going to have an effect on bias, but mainly confidence interval coverage. The constant variance assumption is also something to be aware of as well. So normality and constant variance can be checked in this model using typical regression diagnostics, but that's not something you wouldn't design your trial thinking that you're going to check your assumptions at the end. You would need a good bit of background information, as we had with our clinical collaborators. They knew their biomarkers and their pain distributions very well because they've been working in the area for a long time. So the other major limitation right now is that the development is for simple linear regression only. And so I'll circle back to that in the future work section. But the potential impact, I think, is really interesting. So it can easily be calculated from closed form estimates. And it's also a beneficial sampling scheme, as I discussed, for certain research areas where you have a good idea of what the distribution of your biomarker and your response would be. So because it is easily calculated, it can be a bridge between current ODS literature and clinical researchers that would really benefit from ODS. So outcome-dependent sampling, as the literature is now, it's really hard to break into. It's quite a complex research topic. And so the people who might really benefit from it, as I've seen it, as the literature is, they're not really getting into it. They're not using it. So potentially, if you wanted to get into a more complex ODS situation and you're just trying to get through the literature and understand, I think that looking at this method, looking at a sample analysis with this method can kind of place it in your head and help you decide, okay, where do I need to go from here? Is this appropriate for me, or do I need some more fancy semi-parametric likelihood-based methods? So switching gears, first of all, let me pause. Does anyone have any questions? Okay, so switching gears. So looking at a prognostic model for acute lymphoblastic leukemia. For those of you who don't know, acute lymphoblastic leukemia, or ALL, is a fast-growing cancer of the blood that affects your white blood cells. Leukemia is the most common childhood cancer, and ALL is the most prevalent cancer of the leukemias. So this affects a large portion of patients. So thanks to constantly evolving risk stratification by our clinical collaborators, ALL has really good outcomes. Most individuals can actually be cured, which is incredible. And so when I say constantly evolving risk stratification, here's what I mean. So here's a snippet from the most recent, someone recognizes this? Okay, here's the snippet from the most recent clinical trial that's going on in NCI standard risk ALL. So what our clinical collaborators are doing is they're filtering patients into different therapy arms based on their risk factors. So here we have standard risk average, which is lower risk than standard risk high, and they're getting different treatments. So what factors contribute to those standard risk average, standard risk high? Well, age, cytogenetics, you can be favorable with ETV6 RUNX1 or double trisomies, or you can be unfavorable with a KMT2A rearrangement. You might previously have known this as MLL. You could have IM21 or hypodiploidy. White blood cell count, central nervous system involvement, so whether the cancer has spread to your brain or spinal cord, and minimum residual disease. If you haven't heard of MRD before, my clinical collaborators are going to flinch, but I like to think of it as the remaining cancer cells that are hiding in your blood at a particular time point that it's tested. And so COG typically tests at day 8 and day 29, which is end of induction. So what our clinical collaborators, I know this slide is scary, but what our clinical collaborators came to us with is this, focus on standard risk favorable in the top left corner. So we see that they are assigning people to standard risk favorable if they have CNS status of 1 or 2, which is not a lot of entrance into your central nervous system. So CNS 3 would be the worst. Favorable cytogenetics, day 8 peripheral blood MRD of less than 1%, and end of induction MRD of less than 0.01%. So across the board, we see that they are dichotomizing MRD, both at day 8 and end of induction, when really it could be treated as a continuous risk factor. And I have asked them before, the cutoff is kind of arbitrary in their own words. Yeah, is it ever not? So should we be dichotomizing there? That was one question that they came to us with, and it doesn't just apply to MRD. Age and white blood cell count are automatically dichotomized in NCI status assignment. So they came to us asking, could we do better in our risk stratification if we did not dichotomize our variables? And could we do better, or should we be assigning different weights to these risk factors? And so that might send off a light in your head that says regression model, because we can treat those as continuous and assign different weights. So actually the first investigators to use MRD as a continuous variable in ALL were our UK colleagues. They used the following transformation, which I think is actually an interesting potential line of future work. I'm not married to this one, but it works well in the literature. So MRD is subject to a lower limit of detection. Right now, it's 1 × 10⁻⁵, but that is fluid and evolving, so it will keep getting lower and lower. So what they've done is, if they know that MRD is lower than the lower limit of detection, they are assigning it a value that they call 1 log less than the lower limit of detection. It's just the smaller number than the lower limit of detection, and they're applying an absolute value of the log transformation. So same thing here, if they actually know the MRD value, they're plugging in that MRD value. And this never happens in practice. This is just an edge case to keep it away from 1. But if it were approaching 1, they would plug in this number here. And so what they came up with was a continuous risk score. It is the linear predictor of a Cox model that includes white blood cell count, MRD at day 29, and favorable and unfavorable cytogenetics. So the primary research aims for this project were to externally validate that United Kingdom model in our COG patient population, to develop and validate a new model that was specifically tailored for our patient population if the external validation failed, and to determine and explore model-defined risk groups. Just to give you guys an idea of the data that we were working with, so the newest available generation of COG trials in ALL, the standard risk, the newest available generation of trials, including standard risk and high risk, had 13,200 individuals in it. And so we reserved that as our training data. And the previous generation before that had 7,999 individuals available to us, and we use that as our testing data. Yes, please.
I have a question for COG measure for residual disease in BALL. Is it always via PCR for the expanded B cell clone?
No, okay, so, well, I don't know for sure. I don't think it is always PCR, and that's why I said this is fluid. They are exploring new methods like as we speak. So high throughput sequencing, minimum residual disease is like the next hot thing that they're really excited about. It's not uniform yet, so I can't really use it yet, but that is absolutely something that we need
So
to
the
be.
first ask is that if you have like a pre-B, you would expect to be able to track it, the B cell clone, or something like a clone B, MLL rearranged kid.
Okay.
And something like NGS might have a different limited detection. I'm wondering if that, I'm wondering if conditioning on that has any relevance for the score.
I am not going to pretend to know because that was a little bit technical for me. So NGS.
They see a sequencing like duplex or.
Okay. Okay, that is absolutely something to look into. I think there are so many factors that could potentially improve this model as we move forward and new things are uncovered. So yeah, that is something I will definitely look into.
I think all of the NGS, all of the sequencing based approaches currently have less sensitivity.
Mhm.
So that's what, yeah, I'm wondering about this.
Gotcha. Okay, I will definitely ask my collaborators about that. Thanks.
And just to be
Yeah.
clear on what you mean by the external validation of their model,
Mhm.
it's not just how they define the variables that you're looking for, finding the weights and population. It's actually their specific model with their weights and
Yeah.
their cutoffs.
That's exactly correct. So it's marketed right now as, being applicable to everyone, and I'll show you guys. It does its job. If we needed to use it, I think we could use it with a decent amount of discrimination, but it's not ideal. So yeah. Yes, so 13,000 and almost 8,000. So we had a decent sample size to be working with. Of note, we are excluding Down syndrome patients and Philadelphia chromosome positive patients because they're so biologically distinct. They really need their own study there. So, and this is what David was mentioning. So looking at the existing model external validation, the United Kingdom UK Prognostic Index. So this first bullet is not a part of true external validation, but it is still something you want to look at when you're examining an external model with your own data. Are the directions of effects similar? Are you seeing kind of similar magnitudes? And we did see when we refit the UK Prognostic Index model in our COG patient data that we had a consistent direction of effects, and that's shown in this table, and we're comparing it to their published model equation here. So you might notice that transformed MRD has a hazard ratio of less than 1, which may send off a flag in your head. Hey, shouldn't MRD have a risky hazard ratio, not a protective hazard ratio? And it's just because of that transformation on MRD. A lower transformed day 29 MRD in this case actually corresponds to a higher MRD. So this does indicate that it is a risky factor there. So when we look at the published UK Prognostic Index in our data, the discrimination is mostly maintained. So when they published it, it had a C-index of 0.735, and in our data, it is 0.725. So just a little drop there, not too bad, and still well within acceptable limits for a concordance index. So the calibration slope, which is what we were primarily concerned about, we want our model to be well calibrated, which means that the observed probability of relapse is similar to the model predicted probability of relapse. And ideally, it would be a perfectly straight line where the observed and model predicted probabilities are perfectly equal. So what you're testing for is something called the calibration slope, and that's the slope on the published linear predictor in your data. You want that to be equal to 1. And we did find that it was significantly different from 1. So it's indicative of poorer performance in our validation data than in their development data, which of course we would expect. However, since we have such a large patient population, we can do better in terms of calibration by deriving our own model. It would have been excellent to do additional external validation. Unfortunately, I think probably a lot of you know that this is often limited because you can't get your hands on estimates of the baseline survival function for external models. It's very difficult. Our collaborators were particularly interested in what would happen if we added day 8 MRD to the published UKPI model. This is because day 8 MRD uniform collection is quite unique to COG. I think they're probably the only ones who uniformly collect it. So they were really excited about this. We do see that it significantly adds to the model, but we have a massive sample size, so it's quite easy to pick up on small effects there. We saw a mild boost in discrimination, but the hazard ratio was quite modest, but it did add significantly to the model. So in terms of specifying our own model that is tailored to our patient population, we saw that the Cox proportional hazards model has performed well in ALL historically in the literature. So we felt comfortable moving forward with that. And of course, I'll show you the assumption checks later. But we wanted day 29 MRD, day 8 MRD, favorable risk genetics, unfavorable risk genetics, long transformed white blood cell count, CNS status, and age at diagnosis, which we modeled with a restricted cubic spline using 4 basis knots. And so when we fit the model, you can see we discussed that day 29 and day 8 MRD have that less than 1 hazard ratio, which actually indicates that increased MRD is associated with increased risk of relapse. We see here that favorable risk genetics, as we might expect, is associated with a decreased risk of relapse, whereas unfavorable risk genetics, an increase in long transformed white blood cell count, and a CNS status of 2 or 3 relative to CNS status of 1 are associated with increased risk of relapse. Of course, since age is modeled as a spline, there isn't a single hazard ratio that can tell us what age is doing in the model, but we can visualize the effect of age in the model by fixing continuous variables at their medians and by fixing categorical variables at their reference levels. So here's just an example. We have the log relative hazard on the y-axis, and we have age at diagnosis on the x-axis here. And this was actually really cool to see because it was clinically intuitive. The younger you are, it's really poor with infants, but also it's known that the young kids, they have poor outcomes, and it decreases until a certain point. Here it's showing around 3.5 years old, and then there's a sharp increase, which tapers off around 11, 12 years of age, which they also, I mean, almost anything can be clinically intuitive if you read into it, but they thought that was clinically intuitive because by 11, 12 years old, you're kind of transitioning into being a teenager. So it's tapering off a little bit there. Yes, please.
So the cytogenetic differences are also kind of pronounced. If you stratify by that, are there groups where there's a much greater impact of having day 8 MRD added to the model or it's not?
That is a good question. Let's see. So that I think we're going to cover in, what is this slide? It always take forever. So we didn't look at day 8, an interaction between day 8 and cytogenetics. We looked at day 29 in cytogenetics, and that interaction was not significantly needed in the model. So our clinical collaborators didn't think that day 8 in cytogenetics would be an interesting interaction to look at. So it's not something I took a look at, but that is, you know, absolutely something that we could look at moving forward. So we were trying to be really conservative with cherry picking things that we looked at. We were trying to really pre-specify everything because the whole goal here is that we want it to be externally generalizable to the next patient population. So instead of fitting everything we could think of, we tried to be really, really conservative about looking at items that they thought were clinically important. So anyway, that's just to say, I didn't take a look at the interaction between day 8 MRD and genetics, but that certainly would be interesting. And I think day 8 MRD deserves its whole own paper of exploration because it really hasn't been looked at enough in general. But anyway, that was the additivity assumption. Okay. So the proportional hazards assumption, of course, since we had a very large sample size, plotting the scale of Schoenfeld residuals over time, we did pick up on non-proportionality. However, it is quite sensitive to small departures from proportionality. So we were actually pretty happy with how flat the Schoenfeld residuals were over time. So we were not concerned about the proportional hazards assumption here. Additionally, we plotted the DF beta residuals. So that is the difference in estimated beta coefficient when you remove each individual and refit your model. And so these plots just show you the magnitude of the change in beta for each removed point. And none of the magnitudes were large enough for us to be seriously concerned about influential observations here. Thinking. There we go. We already covered the additivity, but essentially what we did is we considered a pre-specified set of interactions that our collaborators thought were clinically relevant. We tested them as a group. They did not meet our criteria to stay in the model, and so we removed them as a group. However, you know, this was a little close for comfort for us. This was kind of like almost borderline significant, although we have such a large sample size. If it's going to be significant, it's going to pick it up. But this was a little borderline for us. We were a little uncomfortable with that. So what we wanted to do was explore some common machine learning alternatives. I know it's like the thing to do right now, but we just wanted to see if relaxing assumptions could bias anything in terms of discrimination. So are there interactions in there that are important that could have helped us, even though we didn't pick up on it? So we looked at, in the benchmarking study, I compared the Cox proportional hazards model with a survival random forest, a survival nonlinear support vector machine, and a gradient boosted Cox model. What we found is that the Cox model performed pretty comparably to the machine learning alternatives. This wasn't surprising because I think machine learning really shines when there's a lot of information that you don't have a ton of knowledge about, or maybe you have some knowledge about, but you don't know how it's all working together. The people in ALL have been doing risk stratification so much, they have a really good understanding of the variables and how they're working together. And I'm at 10 minutes now, which is not good. So I will just mention that the survival SVM was fit on a representative subsample because it is a new technology and the computation time is quite intensive, so it's not there yet. And I will circle back to this piece in my future work slide. For internal validation, we did a bootstrap-based internal validation of the model, and what we were looking for were low estimates of optimism, which is the difference between your summary metric in that you have estimated and the true underlying summary metric in the population. And so we saw very small estimates of optimism here, which might be expected with the large sample size. And then taking a look at calibration. So to read the calibration curve, we have the fraction surviving 5 years that's observed on the y-axis. And then my bar is here. Okay, well, the model predicted probability of surviving for 5 years is on the x-axis down here. So this is the training sample. We expect a trivially perfect calibration curve in the training sample. So I just threw this in here to preface how to read a calibration curve. what's really interesting is the calibration curves in the testing samples. So 0232 is our high-risk population here, and you can see that there are more individuals with a lower predicted survival probability. So for this high-risk trial, the calibration is actually really good. We might get a slight overestimation in this area of the predicted survival probability where we don't have a lot of data. On the other hand, for 0331, which was our standard risk population, we're getting an underestimation of risk across the board, which tapers. It does taper where we get to areas where we have more data, but this is something to be aware of as we go to apply this in the future. Recalibration and model updating might be needed. So this is just now that we are confident that our model is looking good, we can look at the linear predictor or the risk score from the model. These are the risk scores from the UKPI and the COG PI respectively. We can also plot these within genetic risk groups. So this is the COG PI. And you can see that the risk score is lower in the favorable risk group, followed by intermediate risk genetics, which is not favorable, not unfavorable, and then highest in the unfavorable risk genetics group. The outliers here indicate the other variables contributing to the risk score. We were very happy with the performance in clinical subgroups. So what you're seeing here is the calibration slope. And what we want is that perfect slope of 1. So within all of the subgroups, the 95% confidence interval is overlapping with the beta equals 1 line. So more work certainly needs to be done in non-Hispanic Asian and non-Hispanic other because we didn't have very many events in those categories, so I don't have very much information there. So topic of future work. So to determine risk groups from the risk score, we maximize the concordance probability estimator using the genetic algorithm implementation in the CAT-PREDI package. So we selected 4 risk groups, 3 cut points. We call them low, standard, intermediate, and high. When we plot these cut points, I hope it's pretty visible on the screen there, but we have very good separation between the Kaplan-Meier curves. And probably the most practical implementation. So if we compare these COG PI risk groups with the current COG risk classifications, there are 6 of them as it stands. When the risk is very low or very high, the classifiers are almost in complete agreement. So for example, in standard risk favorable, which is our lowest risk COG group, they're all concentrated in the low and standard COG PI groups. Similarly, for very high risk, all concentrated in the high risk group. So it gets interesting when you have a more moderate risk of relapse. So for standard risk average here, we have kind of a spread, and we have this group of individuals that's doing really well on their current therapy. So it's very important on their current therapy. They're doing very well on their current therapy. And so the clinicians look at this and they say, hey, they're doing really well on their current therapy. Our next move, because we have great outcomes, is to de-intensify. Can we target these guys for de-intensification? It's a very hard question to answer. In my opinion, it requires a lot of validation, and I have a slide on that in future work. Same for high risk, although this is a wee bit easier to answer. There's a group that is doing very poorly on the high risk study on their therapy. And so can we target them with novel therapies? So in summary, we externally validated the existing model. We rigorously developed our own new model that's specific to the COG patient population. And we saw potential to refine the current COG risk classification, but additional validation is definitely necessary. And so in my lines of future work, I hope to conduct that clinical validation on the new COG decision rule. So the best way to do this is with a small cluster randomized trial. That is kind of reaching for the stars, but I'll keep it in my back pocket because the international consortiums are actually really interested in collaborating these days. So it's great, but certainly a prospective multi-center cohort study on the current AALL 1731 Additionally, we need external validation in all race and ethnic minority populations. One of our collaborators has found that there are outcome disparities that are not all attributable to genetics between the race and ethnic groups. As a first start, we have a P20 funded redial consortium that has offered use of their data for validation in Hispanic Latino communities. They have mature outcome data, which is so rare for ALL because outcomes take forever to pop up, and they have biospecimens. Additionally, we need to conduct validation in, I know you can't see it and I can't move it, Philadelphia chromosome like ALL, which hasn't had uniform testing until now. So additionally, I'd like to look into missing data methods for new subject risk prediction with survival endpoints. So Hoogland and colleagues did a really lovely study for binary data, and then they kind of left survival data as an afterthought. I'd like to do a simulation and resampling based study looking at multiple survival distributions and censoring levels to really get a feel for what is the most appropriate method for survival data. And continued exploration for that survival SVM for prediction. I have a collaboration with Dr. Mushon Liang at UF. His idea is to conduct doubly robust learning for the prognostic index under concordance criteria, and he believes that his new objective function can speed up the fitting of that SVM. So I'm hopeful that could be really interesting. And in clinical study design, I would like to extend the ODEB estimation to multiple linear regression, try to relax those normality assumptions, possibly with robust regression. I'm also working on a promising zone design project with co-primary endpoints that's in limbo with our Medtronic partner right now. But in the interim, I'm also working on a design comparing the sequential parallel comparison design and the drop the loser design, which addresses placebo response while identifying the most promising treatment. So that's something that I've been collaborating with a couple of colleagues on that I'm excited about. And we're at time. I'm sorry, there were so many technical difficulties and I only left 2 minutes for questions, but these are my references. And
Question.
yeah.
For the prediction modeling part of your talk, I'm curious if you had, because I've done some similar work before, like working multiple studies and trying to either build or validate. And sometimes I encountered where an entire study wouldn't collect one of the variables I needed, or a large part, or it wasn't collected for a large part of one of the studies.
Okay.
Did you encounter anything like that, or were all of these
Nothing.
data elements pretty much standardly and completely collected across?
So ALL, these studies in particular, it was very uniform. What I did come across was large chunks of day 8 MRD in the older studies because it wasn't collected as often. Some sites didn't have the resources for it. Since that was our testing data and not our training data, it wasn't a huge deal for us, but we still did multiple imputation sensitivity analysis. More work pending that future work on missing data methods for new people, but that's where I encountered that.
And I'm also curious if you ever put these models into like nomogram form.
You know what? I
The
tried that. I like it. My collaborators prefer to Shiny app.
Well,
So
that's it first.
yeah, so I kind of scrapped it a while ago, but I like the nomograms. They're good for visualization for sure.
Very cool.
Did their missingness on the data MRD impact which data set you use, your decision regarding which one to use as your training set and your validation set, or?
No, it was actually more, we knew that the later in time it got, the more similar to future studies it would be. So we wanted to keep it the most generalizable to a future study that we would apply it to. The missing data discussion didn't enter there. Yeah. Although the unintended benefit.
All right, let's take Natalie.